All right, welcome to another week of Full Seminar Online. Before we begin, I want to do a quick little announcement. So every week, Rebecca is hard at work of doing a full seminar online. So I just want to point this out. This ends up going on several different media outlets. So it goes on Twitter, it goes on Instagram, and it goes on Facebook. So you can pick your favorite media and you can check out the full seminar of the week. If you don't have any of those accounts, Twitter, you can view it on Twitter anytime you want. She always does it on Mondays. So call on Tuesdays and you can see what's going on. This is a great way to get involved with the community as well. If you're not quite ready to present work, but you want to highlight a species that you've been working on, you can submit to full seminar online, like Solanaceae of the week. That's on the web page. So I just want to point that out if people are wanting to do this. So you can see here is a Google form. I'll take you to a Google form, fill it out, and Rebecca will contact you. If you know somebody who's doing and takes great pictures, totally submit them and Rebecca will reach out to them. Maybe that might give them the push to kind of submit something. I would also point out, we're starting a plan for spring, and this is all volunteer based. So it's always helpful if you give us suggestions of people who to nominate, or if you feel ready to present, definitely reach out to us and you could submit a speaker or nominate yourself. There's no shame in self-promotion because it also helps us quite a bit. Okay, with all that said, all those announcements, I want to introduce our speaker for today. So today I'm introducing Jill Miller, which is, she's a professor at biology and Thomas F. Crick Reader in the environmental studies at Amherst College. Jill Miller got her undergrad at Colorado College. She then continued on to do her PhD work at the University of Arizona. She then did her postdoc work for a short stint in Solanum with Pam Diggle at the University of Colorado Boulder. And now she's currently at Western Massachusetts working at Amherst College. And with that, you can share your screen, Jill, and go ahead and get started.
Okay, everybody see that? Okay, actually I need to do one thing. I apologize. I need to get rid of that. Okay, so thank you very much. I'm actually really excited about the opportunity to present my work. I've have a couple of new irons in the fire, and I'm really wanting to talk about that. For those of you who don't know, Amherst is a very small town in Western Massachusetts. It's about 2 hours from Boston. It's about 4 hours from New York City. Amherst College is an undergraduate institution, liberal arts undergraduate institution. So the main collaborations I have are with undergraduates who are working on their research in my lab. Okay. All right, so most broadly here, my research interests are on the ecology and evolution of plant reproduction, and I've spent much of the last 20 years or so working in the plant genus Lycium. So you can see some species of Lycium here on this slide. And I hope today that I convince you that Lycium is a pretty nifty group in actually to study reproductive evolution. I'm going to start by sort of trying to distinguish or define two different features that I'm going to be talking about. So for example, I'm going to talk about mating system transitions from self-compatibility and that continuum between self-compatibility and self-incompatibility. So transitions where we're losing incompatibility and going back to compatibility. And my lab is quite interested in the genetics that underlie the self-incompatibility systems. And like the Solanaceae as a whole, the genus Lycium is in the self-incompatibility systems in Lycium are encoded by several linked genes in an sRNA-spaced non-self recognition system. And so we'll talk a little bit about those genes and their evolution in this transition. And then the second thing that I'm quite interested in is how sexual function gets packaged, both either within a flower or between flowers on different plants. And so for the vast majority of Lycium species, you have what I call co-sexual populations with hermaphroditic flowers. So we're looking right here at a Elysium pallidum. You can easily see the male and the female sexual structures, or Elysium pharisissimum has that same pattern. So that's really what you see in Lycium when sexes are combined within an individual. But you also see the evolution of dimorphic strategies or separated sexes. We might have, in this case, a morphological hermaphrodite and a male sterile plant. So these are flowers from different plants. Or in this case, you have a male plant. There are no female structures in here, but you also see females. So you either have gynodaceae or dioicae in Lycium. And I'm really interested in what the evolutionary forces are that cause these transitions and how frequently they occur in Lycium and how they intersect with other particular traits we might be interested in. And then finally, I went to the University of Arizona, and my research is really very much rooted in a phylogenetic perspective. So the historical or comparative approach is being really important to understanding these evolutionary transitions. So I'd like to just spend the first few minutes of the talk providing background about what we know about reproductive evolution in Lycium, and then I'll focus in on the particular cases that I'd like to talk about. So Lycium is a moderately sized genus in Solanaceae. It has about 92 species, something like that. It has a very widespread distribution. So it's found on every single continent minus Antarctica, but it has three main centers of species diversity. So you find a lot of species in Southern Africa. You find a lot of species in Argentina and Chile, and then you find a lot of species in the desert Southwest in the US and certainly down into Mexico. The distribution worldwide tends to be subtropical. There are a few exceptions, but it's mostly a subtropical distribution. And the plants are pretty drought and salt tolerant. So here's a Lycium from the Sonoran Desert. Here is a Lycium that is coastal. And so that's the other thing you find. If it's not in a dry, semi-arid place, it might be coastal. So it seems very likely that they are quite salt tolerant as well. And when you look across the group, you see quite a bit of floral morphology, particularly or floral diversity, and you also see this with regard to sexual function, which I'm gonna talk a bit about. So I have a long-term collaborator, Dr. Rachel Levin. She is also at Amherst College, and she and I, over the past 15 years or so, have really tried to work out evolutionary relationships across species for a worldwide sample of Lycium. And so just to orient you, we've sampled about 80 of the 90 species. We've used a battery of different approaches, and this tree is just summarizing some of the things that we've learned. So there's pretty good regional structure in Lycium. There are no shared species, for example, between North and South America. But we see a really clear pattern in these data of a single dispersal out of the Americas and into Africa and Asia. And so we have good support for monophyly of all of those taxa collected from that part of the world, but really poor resolution at the base of that tree. So we know it's a young genus. We know it's dispersed out of America a single time. And we know that Lycium also sort of gets around. So you can find Lycium on many different islands or in island archipelagos. So here's a Lycium from Hawaii. This is a Lycium minimum here next to this marine iguana. And then this is a Lycium from the Channel Islands in California. So we know it's moving around, it has these, if you know goji berry, that's also a Lycium, right? So it has these multi-seeded fleshy berries for the most part that enable, we think, this dispersal. So these data are all good and well. We have a new approach in the lab. We've been taking a rad sequencing approach to try to get a little bit more variation among this group to sort out these relationships. So we have a smaller data set with only about 58 taxa, but we have SNPs across them and about 500 KB, 500,000 base pair alignment here. And so we can look at relationship here, and we largely confirm what we've already knew, which is we have a single dispersal out of America. We have frequent dispersal on islands, but relevant to today's talk, this particular analysis had more sampling of dimorphic sexual strategies. And what you can see is that dimorphism seems to evolve multiple times in multiple places all over the tree for Lycium. So we know these sort of general large patterns when we look across species. And the other really important trait that I want to introduce here is to suggest to you that there's this kind of a curious pattern when you look at sexual system variation. So variation between whether or not plants are cosexual or dimorphic and what their chromosome number is. And the short story is that polyploid Lycium species are always dimorphic, And diploid species tend to be mostly cosexual. And so we have this coupling of traits with, and by cosexual, of course, I mean hermaphroditic sexual expression, that always pairs with diploidy, whereas any kind of dimorphism, whether it's gynodioecy or dioecy, pairs with polyploidy. And so I was looking at these, this is back from my dissertation, I'm looking at these traits and I'm trying to understand how they might be connected across these species. And I know that Lycium is part of this big Solanaceae X equals 12 clade. So I know that the base number of chromosomes here for Elysium is 2 and 24. I also understand that dimorphism is quite rare in the Solanaceae. It's pretty frequent in Elysium, but it's pretty rare in the family. And when dimorphism evolves, you can always find these abortive anthers. And so it's pretty clear that dimorphism is evolving on a background here of hermaphrodites. And then finally, my lab has done more recent work on these self-incompatibility systems. And we know that in the family as a whole, and also in the genus Lithium, that self-incompatibility is an ancestral trait in the genus. And so I'll show you a little bit of data later to suggest that's true. So I'm looking at these patterns and I'm trying, 20 years ago now, I came up with a pathway to try to explain what might connect this side where we see species and this side where we see species. And the idea here was pretty straightforward. It's simply that you have a polyploid event that disrupts incompatibility, and there's quite a bit of empirical evidence to suggest that that could happen. So you have a transition from diploidy to polyploidy, or from incompatibility to compatibility. And then you've got populations that look like this, which of course, because self-incompatibility is ancestral, have never experienced selfing, and we would expect then, if they are selfing, to express pretty high levels of inbreeding depression. And we think that might be the trigger, this selfing and inbreeding depression, to make a population transition from being cosexual to being dimorphic, so that dimorphism here evolves as a secondary outcrossing mechanism. Now, I emphasize that this pathway was me looking across different species of Lycium and trying to understand what might connect the two. What I want to do today is slightly different. What I want to do today is talk a little bit more about within species variation for these same traits. So we've got single species that are varying in sexual strategy, in ploidy level, and in mating system, and try to leverage that within species variation to actually continue to address and investigate questions regarding reproductive evolution. So this is sort of the backdrop of where I started when I started my position at Amherst about what I knew about incompatibility, or pardon me, about this system. And so what I want to do today is actually talk about 3 taxa that actually have a lot of what I think is pretty incredible variation, and the way that variation intersects with their reproductive biology and evolution. So for example, we'll start here with Elysium Carolinianum, and we're going to think here about how the phylogeographic history of this taxon intersects with its reproductive biology. And then I want to transition to just thinking about floral size changes and try to use this within species variation to address how we could characterize flower size changes after the evolution of separate sexes. And then finally, I'm super excited to tell you about Elysium australis. So our latest trips have taken us to Australia, and how that variation in sexual expression and ploidy actually contributes to the potential for reproductive isolation in that group. So this is sort of the outline for the rest of the talk. Let's jump into Elysium Carolinianum. So Elysium Carolinianum is this interesting species that has a really interesting distribution. So again, you're looking at the distribution of the genus as a whole. Anything in colors where you might find Elysium. Elysium Carolinianum's mainland range is along the Gulf of Mexico here. So you can find it all along the Gulf of Mexico, in Florida, through Louisiana, through Texas, and down and up into the Yucatan. It's probably in Cuba as well. You also can find it on the Atlantic coast, but there's only one species, and that's Elysium Carolinianum, that has this eastern range in North America. But you also find Elysium Carolinianum in Hawaii, and in the southern hemisphere in Easter Island, and south of Japan. So these are massive distances. It's pretty amazing to find that distribution, one of the most widespread species in the group. So I want to think about reproduction and reproductive evolution in the context of that distribution. So in this species, we've taken a genotype by sequencing approach. So we have sampled 120 accessions across the range of Carolinianum. So from Florida to Mexico to Texas to several different chains in the Pacific. And we've used, again, the IPyRed pipeline, and we have about 25,000 SNPs. And we've, in this particular tree that I'm showing you, we have analyzed the entire alignment here using maximum likelihood. And what you see in this species is incredible regional structure. So you see the Gulf Coast and the Atlantic in Florida, populations in Mexico, populations in Texas, several different populations in Texas. You see the two different islands in the Ogasawaras. You see the different islands in Hawaii, and these actually have relatively strong support. I just can't fit them in on that branch, but it looks very clearly like this species has gone out into the Pacific a single time, right? So dispersed a single time out there. We've done a number of different kinds of sensitivities on this. We've also just analyzed the SNPs using a coalescent base approach. I've just color coded this so that you can see that you generally get the same patterns. You pick up the same kind of structure if you use that approach as opposed to this likelihood approach. And we've also done some standard sequencing. So we have about 9,000 base pairs of sequence data from plastid and nuclear regions, with using more outgroups here to really just assess the monophyly of Carolinianum. So that's actually pretty important. And I should mention that many people actually refer to Elysium Carolinianum in Hawaii or in Ogasawara or in Easter Island as Lycium variety sandwichense. And so it's clearly something, it's gone out there a single time, but it's also very closely related to other members of Carolinianum that are in that mainland range. So we know a lot about its phylogeographic history, and this, I think, coupled with the idea that incompatibility is ancestral in Lycium, led us to ask the question, well, what do mating systems look like across the range of this taxon? So we were really quite interested in thinking about mating system transitions potentially in this group. And so these are really straightforward experiments, right? So we're either going to mark buds and leave them open to pollinators to get control levels of fruit set, or we're going to keep them bagged and see if they can set fruit or seed autogamously. But our most important thing that we're looking at here, if we're talking about mating systems, is whether or not they can successfully set seed following self-pollen. So we had a self-pollen treatment and a cross-pollen treatment, kept those flowers, always emasculated those flowers, and always kept them bagged to make sure that the pollen source was what we wanted it to be. And when you look at data from Texas or from Florida, what you see is a really pretty clear pattern of strong self-incompatibility. So pollen, when the source was self-pollen from the same plant, the plants aren't setting fruit. The data look the same for seed production, whereas cross pollen was very successful in setting fruit and seed. Now, when we think about a long distance dispersal, and Lycium on the whole is probably 5 to 10 million years old or something. And so when we think about these dispersals out to the Pacific, it's complicated to think about how a self-incompatible species could make that transition. And when we went to Hawaii and we did crossing studies in 2 islands in Hawaii, on Kauai and on Maui, what we find is the loss of self-incompatibility, right? So it's very clear that the self-pollen treatment is as good as the cross-pollen treatment. There is no difference there. And so clearly we see within this single species, which is probably not that surprising given the phylogeographic history, the loss of incompatibility with a long distance dispersal. And so I mentioned that we also were interested in the genes that underlie self-incompatibility. And so while we were out doing these pollinations, we also collected material to isolate the genes that underlie and encode incompatibility. So to do this, you just collect styles, toss it in RNA layer, and then go back and use primers to isolate these sRNase alleles. And so in this dashed box, this just shows our sampling across these different populations for these style samples. We brought them back, and we're able to isolate 41 different Lycium Carolinianum sRNase alleles. So this is one of the genes that controls the female side recognition. And when we look at that analysis, so basically we take all the terminals that you see on this tree at the right, are actually alleles at the sRNA locus. And I've pulled the terminal names off if it wasn't a Carolinianum allele. So you should see 41 different Carolinianum alleles organized in and highlighted here by these little yellow circles with letters, which I define as something called a transgeneric lineage. And so I've pulled out a few of these to look at. So here's lineage C. And in lineage C, when we blow it up, we see this is the fourth allele that we identified from Elysium Carolinianum. It's actually most closely related to the 14th allele that we recovered in another species, Lycium parisia. But both of those alleles together are most closely related to this allele that we downloaded from GenBank with a lot of homology here called from Nicotiana lata, which means essentially that this allelic lineage predates the diversification of Lycium and Nicotiana. So it's a very old, ancient lineage that's been retained through negative frequency dependent selection over many millions of years. You can see the same kinds of patterns. So every time you see a little yellow circle like this, it's some case where there's a Carolinianum allele that's more closely related to an allele pulled out of a different genus as opposed to a congeneric allele. And this is the pattern that you'd expect to see if sRNA alleles have been maintained via self-incompatibility over very long timescales. So we weren't terribly surprised with this result at all. But what we were really interested in here was we know we have incompatibility in these two populations, and we know we have self-compatibility here. And so we wanted to see, well, what's the consequence of those transitions on allelic diversity? And we can think about allelic diversity here in a couple of different ways. We can either think about the number of alleles that we recover in a population. So for example, we sampled 23 individuals from Florida and we recovered 8 different alleles. Those 8 alleles were in 5 different transgeneric lineages. So I'm just showing you in a pie chart the proportion of those 8 alleles that were in a particular lineage. And over here, I'm showing you which lineages were present in the Florida population. And so if we look at our mainland populations, so if you look at Texas, there was a whopping 21 alleles present in this Texan population, and it had many, many, many transgeneric lineages. So when you look at this tree, all the colors that you see in the tree on the right are lineages of alleles that are retained in these self-incompatible populations. So the question here is we know we have a transition to compatibility. What does it look like then when you sample sRNA alleles? Can you even recover them in these Pacific Island populations? And the answer to that is yes. But what you recover is pretty interesting, right? So we've recovered only 6 total alleles, and they're all restricted to one of 2 lineages. So lineage P or lineage B. So you've bracketed them here. So these alleles are recovered from Hawaii, Easter Island, and Ogasawara. And these are very, very similar to each other. And so what you see is the loss of this sRNA diversity that's happening, of course, at the same time that self-compatibility is evolving. There's a couple of mechanisms by which this might occur. So the first would be bottlenecking as plants are dispersing out into the Pacific, right? We don't imagine that that's a very common event. And so whatever those initial colonists had in terms of their sRNA diversity is sort of what the baseline Pacific populations had to work with. So it'd be interesting to sort of try to understand what the initial bottleneck looked like. But it's also the case that there very likely was selection for self-compatibility in the establishers on the island. And so whichever sRNases were left in those individuals that ultimately became self-compatible are also very likely to spread. So we have a couple of different mechanisms here to think about that loss of sRNA diversity. So kind of unbelievably in the system, what we've also noticed, so let me just back up before I tell you the rest of it. So we've got these self-incompatible mainland populations and self-compatible island populations. And we've confirmed that with crosses and with RNA diversity patterns. But the other thing that we see is in this species, you typically have all plants produce these hermaphroditic flowers. That's the most common phenotype that you see. But we do have the evolution of dimorphism here, and it's happened actually twice in this species. So it appears that Gynodiaceae has evolved both in Maui on Hawaii and also in Mexico. And so these pictures here are from a population in Maui where you can clearly see abortive anthers here. This is the female mating type. And then you can see in the same population, a hermaphroditic plant. And so you've got Gynodiaceae in some populations on Maui, not even all populations, but only some. And then you also see the same pattern in Mexico, the same evolution. So let me just show you a little bit of what this looks like. These are the 3 populations in Maui that we know have evolved sexual dimorphism, or pardon me, gender dimorphism. And what you can see are the abortive anthers here in flowers from the female mating type, whereas you have a nice, long, well-developed stigma. This is the male mating type, or the hermaphrodites, shown for Lapoa, and the hermaphrodite morph is always larger. That's what this axis is showing here in Hawaii. Again, you see that same pattern in the Yucatan, only the flower size dimorphism is clearly more obvious. So flowers from female plants are much smaller, considerably smaller than flowers from these hermaphrodite plants. So whenever I'm trained now, whenever I see the evolution of Gynodiaceae in Lycium, I'm trained to look for polyploid, polyploidy, or for dimorphic associations with dimorphic sexual expression. And so we used flow cytometry across the range here to actually assess what the ploidy levels were in Maui, in the Yucatan, and in the mainland populations. And what we found was that all populations in Hawaii, it doesn't matter if you're in Kauai, Oahu, or Hawaii, they're always diploid. And even dimorphic populations in Maui are diploid. So we recognize here then that Gynodiaceae is evolving on a background of diploidy. But in the Yucatan, all the samples that we used for flow cytometry came back to us as tetraploids. And so in the Yucatan, we have a different pattern where you have a polyploid event that's associated with the evolution of dimorphic sexual strategies. We, of course, are very interested in sort of the baseline or the evolution, the flow cytometry measurements in these Texan and Floridan populations. It turns out those are also diploid. And so clearly we've got one polyploid evolution here, but we've got convergent evolution of dimorphism. So that's a whole lot of change in one taxon that's happening over this pretty widespread range. And so let me take you back to this original model that I was thinking about when I was finishing my dissertation and trying to understand the relationship of diploid, hermaphroditic, self-incompatible species with polyploid, dimorphic, compatible species. And if I think about that and I use the within species variation in Carolinianum to try to understand that, what I see are some different things. So I see the presence of diploid, co-sexual, self-incompatible ancestors, if you will, and I understand that the loss of incompatibility, so this transition is actually associated with this oceanic dispersal, right, and establishment in Hawaii. So we've got the loss here without the polyploid up here. We've also shown that inbreeding depression in the hermaphrodites in Hawaii is quite high, and certainly high enough to drive the evolution of a dimorphic or outcrossing population following that. So in Maui, at least in Carolinianum, it's slightly different from that model. If we think, though, about the Yucatan, the pattern in Mexico, we again see this association of polyploidy with dimorphism and diploidy with hermaphroditism, but we didn't have any good field data telling us about the compatibility status of the populations in Mexico. We've been pretty lucky to figure out how to get this thing to flower in our glass houses, and so we've now actually have a little bit of preliminary data suggesting to us pretty strongly here that this thing is actually self-compatible, whether or not you look at fruit set or you look at seed number per fruit. And so we're pretty excited about these data. So if I summarize, or if I try to summarize for you what I think is happening in this single taxon Elysium Carolinianum, what I'm looking at here are multiple triggers that lead to mating system transitions. So multiple triggers that lead to the loss of incompatibility, but then sort of the convergence of the solution to that problem in terms of the evolution of dimorphism in two different lineages in that taxon. So that's the first story I want to tell you about within species variation. If I transition now, this is a quick story, but I want to focus in on floral size changes. And we're going to stick with Elysium Carolinianum because I've just explained that transition to you. And then we're going to add on another species called Elysium Californicum. So just to remind you, in Maui, we've got co-sexual populations where you have hermaphrodite flowers, and we have dimorphic populations where we have the evolution of male sterility or females and hermaphrodites. But we also see this same pattern in a different species called Elysium Californicum. We see hermaphroditic co-sexual populations, and we see dioecious populations that are present. This is the range. This map shows you the range of Elysium Californicum. So it's Channel Islands through Baja, along the Sonoran coast, and then a pretty disjunct and interesting populations out here. Where you find dimorphism in this species is either in the mid-peninsula part of Baja, in its northern part of its range in southern Arizona, or at the very tip of Baja. And not surprisingly, at least in Lycium, when these populations in Elysium Californicum are dimorphic and have males and females, they're also tetraploid. But when these populations are hermaphroditic and co-sexual, they're diploid. So we've got these two different cases where we have the potential here for changes in flower size, which is what I want to talk about after the evolution of dimorphism. So how does floral morphology change? And it turns out that if you look in the literature across lots of species, flowers on pollen-bearing plants, so hermaphroditic flowers in dimorphic populations that are gynodioecious, or male flowers in populations that are dioecious, tend to be larger than their conspecific female plants. And so that's a sort of general pattern. The pattern is really, really clear for gynodioecious taxa. It's a little bit less clear for dioecious taxa, but it's still a pretty strong pattern that you see. What I want to think about is how those differences in flower size evolve, right? It could be that there are that pollen-bearing plants are getting larger, right? The flowers are actually becoming larger. I mean, as soon as you have the evolution of male sterility in these populations, there should be selection on the pollen-bearing plants to evolve better pollen export or higher pollen attraction so they can fertilize those females in that population. So that's an adaptive explanation. But what the central point here is that it involves these guys getting larger. You can also imagine, though, the flip side of that would be, well, maybe it's actually that female plants are evolving to be smaller, that flowers, they're taking the resources that they recover by not making very large flowers, by not making expensive pollen, and they're putting that back into increased seed number. And so there's actually, in that case, it's that females are getting smaller, not males getting larger. You can also imagine some developmental sort of non-adaptive hypotheses. So pollen has a lot of hormones in it, and when plants stop producing pollen, those hormones are not present, and those hormones actually lead to developmentally smaller flowers. So there are all these there's support for all of these hypotheses in the literature. But the thing that I want to emphasize today is that we can use these within species comparisons to actually answer the question, well, did males get bigger? Did females get smaller? And so the way that we do that is to try to find the relevant comparison within taxa. So for example, here's our flower size axis. This is the global average flower size for females in dimorphic populations in Hawaii. And again, flowers on these hermaphrodites are significantly larger than those. If we look across Elysium Californicum, here are females, here are males. Our y-axis is again just flower size. In every population that you find, you see much larger male flowers than you do on flowers from females in those populations. But there is Ploidy variation, which is confounded with dimorphism and comparisons back to these guys. And there's also a bunch of latitudinal variation in flower size across the range of Californicum that we needed to deal with. And so one strategy that we had for doing this was to just restrict our comparisons to sort of co-occurring co-sexual and dimorphic populations. So if we just restrict our comparison to the mid-peninsula of Baja, then we're going to lose a lot of the latitudinal variation that we see, because flowers in the north tend to be larger than flowers in the south. And when we do that, we have 3 dimorphic populations that we have in the mid-Baja and 7 co-sexual populations. And it turns out that when you do that comparison, it's very clear that flowers on male plants are larger. So flowers here have become larger as compared to their co-sexual hermaphrodite conspecifics, and that female flowers are actually just as large as these hermaphrodites. So it really does look like we can sort of eliminate the hypothesis that female flowers are getting smaller for Californica and try to focus in on hypotheses that suggest, well, why would male flowers become larger? So one can go back out to the field and have a look at that. We can do the same kind of comparison in Hawaii for Elysium Carolinianum, and we can, again, we have 3 dimorphic populations and 8 co-sexual populations. And what you find are, this is the flower size on hermaphroditic plants. And what we find is that, in co-sexual populations and flowers of hermaphrodites in dimorphic populations are again significantly larger than their conspecific hermaphrodites in co-sexual populations. So you can see that change. And then you can also see that female flowers are not significantly different from those conspecific hermaphrodites. So again, this within, my argument here would be that this within species variation lets you get at finding the relevant comparisons and finding the most powerful comparisons to actually try to figure out how these traits, these reproductive traits, are changing in real time. Okay. Last story I want to tell you about is our most recent work in Australia, and I'm super happy to present this data. So Lycium australe is the only species of Lycium that is endemic to Australia. So there are a number of different species of Lycium in Australia, but most are introduced. But we believe that Lycium australe is dispersed from southern Africa. It has a really widespread distribution. So if you go to the Australasian Virtual Herbarium and you download all the specimens and then sort of work through them and confirm which ones are truly australe, you find what I'm sort of showing you here in gray. It's big continent, big distribution. And I first met this species in 2011 while I was in Australia for the Botanical Congress, and was out here in the eastern part of the range and saw what I thought looked like male sterility in these populations. And so I got a little bit interested in this species. On that trip, I had the really great fortune of meeting David Simon, who taught me how to find this species. And he said, Jill, what you do is you find these salt lakes, and Lycium will never be on the edge of these salt lakes. But if you walk back from the salt lake up into where it's a little bit more coverage, where you have a little bit more tree coverage, once you get to these eucalyptus coverage, you find Lycium. And so here's a Lycium, 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 Lycium. It just becomes a major part of the understory of these pretty open parts. So it looked like it might have been male sterile to me. And then I started doing a little bit of reading about it. And I noted that Leslie Hagee, who had worked on Lycium in Australia, suggested that specimens in Western Australia out here were somewhat larger or more robust. They had bigger flowers, they had bigger leaves. And of course, when I see male sterility, I think polyploidy. And when I read this sort of more robust plants, I think also polyploidy. So we took a trip from Sydney to Perth and collected as much Lycium as we could get our hands on. And we collected a number of different things. So samples for assessing Ploidy variation, lots of floral samples when the populations were flowering, leaf tissue for genetics, et cetera. This is just an example of the kind of variation I would see when I was out sampling across these populations. And so the first piece of data I want to talk about is Ploidy variation in this taxon. So we sampled about 167, it looks like 167 individuals across this range. And when we do that, we find really neat pockets, so very tidy little modes of DNA content. So this is picograms per 2C is the unit of our DNA content here. And we have a peak at about 5. We have a peak at about 9 and a peak at about 13. And no plants are falling with intermediate DNA contents. Last year, we published a paper where we documented that for this species that have the diploid chromosome number that you'd expect in Lycium, those 15 plants firmly fall into this peak. And so we're pretty confident here that this is our diploid peak, this is our tetraploid peak, and we are calling this peak out here our hexaploid peak. And if you take this ploidy inference and you map it back onto the distribution, what you find is that diploidy is very widespread. So it's very common in the east, all through the Nullarbor and into Western Australia. And that tetraploidy is most common out here in Western Australia, with a couple of isolated pockets, so a couple of populations where we recovered tetraploidy out here in the east. And finally, that those hexaploids are restricted up here in the Shark Bay region. If you're familiar with Australia, this is where the stromatolites were. So basically, you're looking at Elysium Austral and you're looking at stromatolites. So that was a pretty awesome experience. Now, while we were looking at Ploidy variation, we also started to think about, well, let's think about genetic structure across this pretty wide range. And so for these data, we settled on a single plastid marker to start with. It's a great project for a new student undergraduate working in the lab. And so we have 724 individuals that we've got the plastid haplotype for the TERN-H PSBA region. And what we find here are 12 different haplotypes in Lycium Austral, so 12 unique sequences here, and each haplotype is given a number here, so 1 through 8, 9, 10, and 11, and then a 12th haplotype, that was the last one we found. But the vast majority, of course, the size of the circle in this haplotype network references the number of individuals in it. The vast majority of individuals are either in haplotype 9, so there's 281 individuals, or in haplotype 8, which is 331 individuals. So we have lots of individuals in these two main groups. And I think you can see from this, there's not a whole lot of variation. It's a plastid region, but 1, 2, 3, 4, 5 SNPs and a 6 base pair indel separate haplotype 9 from haplotype 8. And we sort of have just designated these group A haplotypes and group B haplotypes. And in the map here, I've shown you in color where you find group A haplotypes, which are restricted to Western Australia out here, and group B haplotypes, which are in Western Australia, at least the southern part of it, and extend all the way over into the east. So remarkably, our ploidy distribution is somewhat consistent with our plastid haplotype distribution, which turned out to be really fortuitous and kind of interesting. And so we wanted to ask the question, well, what is the association of any, if any, of ploidy, so 2x, 4x, 6x, and haplotype at the TERN-H to PSB locus? And what you see is that group A haplotype individuals, so these are plants that have either haplotype 9, 10, 11, or 12, are never diploid. They're always polyploid, and they're usually tetraploid. In contrast, group B haplotype plants are almost always diploid, with the exception of these two pockets out here in the east. So this kind of nifty association that we have. So the next thing we do is we were collecting flowers across this range, and we collect the flowers, and then we had students go through and look for the presence or absence of pollen and primary sexual features. So if you dissect flower buds that are pretty mature and ready to go and then stain and look for pollen, in this sample of 191 individuals, we had pollen in 100 individuals, and we had pollen that wasn't present in about 90 individuals. And it won't surprise you if I tell you that plants that don't make pollen have not very wide anthers and not very long anthers, and that's true regardless if you're a diploid or a tetraploid. It got more interesting, and we can look at those morphologies here. So in the middle here is a male sterile plant. You can see this is a mature flower. This is the size of her anther. This is a millimeter for scale versus a tetraploid plant, much larger, versus a diploid plant over here, much larger. It got more interesting when we looked at female structures. So male sterile plants, diploid male sterile plants have long styles, and diploid plants that produce pollen also have long styles. The same pattern is true for stigma width. So the pollen-bearing morph here looks like it has functional female structures. The same is not true when you look at these tetraploid plants in red. There's females here, they have long styles and wide stigmas, but the pollen-producing plants have actually incredibly short styles and very sort of malformed stigmas. And you can see that here. This is the style, the entire style length of this tetraploid plant, whereas the style of this diploid pollen-bearing plant is much higher. So our interpretation of these data right now is that we seem to have gynodioece, or the presence of females and hermaphrodites for the diploids, but in the polyploids, in the tetraploids, we seem to have females and males, all again within the single taxon. So we wanted to do a little bit more with regard to genetic structure. So we took a rad sequencing approach here. We have 42 individuals, again, across this range. We used an iPyrad pipeline here. We were very stringent. We only kept the 16,000 SNPs that were present in 90% of those 42 individuals. And I did that because I wanted to use structure, and structure, of course, is pretty sensitive to missing data. So when you look at structure plots, and I'm just showing 4 different clustering values here from 2 to 5. And the things to pay attention to in this is these are just genomic numbers of plants. This is sort of meaningless to you, important to me, but what you do need to know is this is the western part of the range and this is the eastern part of the range. So I've just lined them up along the x-axis like that. These are group A plants, so haplotype A, or haplotype B. And it doesn't really matter which cluster you look at, you can totally see that there is a pretty unique clustering of these group A and group B plants. And I can tell you that there are some tetraploids included in here, but they really are falling out with group B plants. So at least it have a couple of origins here of Ploidy for sure. The K=5 cluster got kind of interesting because when you look at that, there's a group of plants here. Actually, all of these plants are in the Western Australian part of the range. In fact, where they're co-occurring with these tetraploids, because you do have co-occurrence out here. And these 3 plants in particular are actually in the same population as diploids. And so this level of structure, whether it's real or not, is hard to say, but it does sort of suggest the possibility of some introgression between these, some sharing of genes between these diploids and these tetraploids. So our latest trip to Australia involved going back out and trying to find mixed populations with both diploids and tetraploids in them. And we were pretty successful at finding a couple that were in really good shape for crossing studies. This is just an overhead map trying to emphasize for you really how close these plants were co-occurring. They're meters from each other in the same population. Luckily, Rachel and I could tell the difference between tetraploid plants and diploid plants, which we confirmed after we got back with flow cytometry. We did 2 kinds of crosses here to try to get at reproductive isolation. We did homoploid crosses, which involved crossing a diploid plant with another diploid plant in the population, where we used a 4x dam and we crossed that plant with a 4x male. We also did the reverse, where we would take a diploid dam and cross that plant with a tetraploid, or take a tetraploid dam and cross that back to a diploid. So we did these crosses and then monitored fruit and seed set, etc. Now I'm showing the data here for one of the populations. I'm going to show you across the top. The second population I'll show you along the bottom. Control is just simply the level of fruit or seed number in populations that were unmanipulated, or in plants that were unmanipulated. And this, of course, is our within Ploidy cross and our between Ploidy cross. So if our dependent variable here is fruit set, what you notice is that in diploids are setting high levels of fruit, regardless of whether or not it's a within Ploidy cross or a between Ploidy cross. So there's no difference here in fruit production with the unmatched pollen, if you will. And that's true in both populations for diploids. That's also true for tetraploids in both populations. Fruit set was higher for tetraploids in Hurlstone, but these again are not differing in terms of their fruit production following these heteroploid crosses. When we look at seed production, we see a slightly different, we see again, no significant difference between sort of this seed number for within and this seed number for between, even though diploids tend to make a few fewer seeds with between crosses. The opposite pattern is actually true for tetraploids, who make a few more seeds with between ploidy crosses than they do within ploidy. And so you see that same pattern actually curiously in both populations, but no overall significant difference between homoploid and heteroploid crosses. So I was scratching my head. I was sitting in Australia. I brought these seeds back and I gave them all to Derek Schneider. He's my undergrad, fabulous student. And I said, Derek, do something with these seeds, germinate these seeds and let's see what's going on in terms of reproduction. And he did. And finally, we found reproductive isolation between the diploids and the tetraploids. And so diploids and tetraploids, these populations, when you have a within ploidy cross, they're germinating about half of the seeds that they're producing. Okay, but when that's a between ploidy cross, it's less than 1%. So there's really strong isolation, reproductive isolation between the diploids and the tetraploids, but it's acting a little bit late. It's acting at sort of a triploid block stage with regard to seed germination. So there's a variety of different questions that I'm still interested in with in Lycium austral that we need to try to understand. We're currently using sort of a niche modeling approach to try to understand the evolution of the distributions of the diploids and the tetraploids. I'm really quite interested in the extent of mixed populations and the stability of mixed populations. One would imagine that one ploidy type should outcompete the other and you shouldn't have these mixed populations. So that's kind of an interesting problem to think about. I don't know. I know nothing about the sexual expression of these tetraploid types out here in the right, unfortunately. And then finally, we're left with some really pretty fascinating questions, historical questions about how changes in ploidy have happened and how sexual expression has changed in this species, particularly because we think that it dispersed from Southern Australia and populated Australia. And so we'd like to understand a little bit more the placement of Australia in the context of those sexual features of the original colonists. And so with that, I will leave you, I think again, my colleague Rachel Levin, some students who really pushed different parts of these three stories that I told you out, as well as a whole bunch of folks who have supported me and supported this work over a long period of time. So thank you very much for your time.
Awesome. Thank you so much, Jill. It was so interesting. So many cool aspects of everything. If you guys have a question, you can either type it out, or if you can raise your hand as well, try to pay attention to that, or you can just type that you have a question. I guess I can kick stuff off. So I think it's actually really interesting that you have like this idea of like how, or a hypothesis how things could happen, like polyploidy to have that first, and then just SC happen first, and then you showed like it could happen both ways. But I was wondering, in your first example of Lithium Carolinianum, like you had the example of like the one in Mexico, it went to SC before, or like there's no ploidy difference in it. Do you think there's more of a mix because there's no reproductive isolation, or do you think it's another means of reproductive isolation that's occurring and not just like this ploidy level variation? Does that make sense?
So I think so, but I think maybe, all right, so in Carolinianum, the Mexican populations are polyploid.
Oh, okay.
And so in that, so again, in that taxon, I'm sorry, I stopped sharing, but I could share that again and show you. Let me see here. So okay, so the Mexican populations that we looked at in the Yucatan are actually tetraploid.
Oh, okay. Is it the other way then?
Say that again.
Hawaii. Is it the other way then? Is Hawaii?
In Hawaii, they're diploid.
So do you think it's like, oh, sorry, go ahead.
No, it's fine. Go.
Okay, I was asking, so do you think it's like just the range isolation, or do you think there's more, I don't know, do you see more co-sexual species within the mix of your dimorphic or co-sexual individuals as well as dimorphic because there isn't reproductive isolation, or do you think like it quickly transition and you don't see a mix of those? Does that make
Okay,
sense?
so let me back up to, for this population, for the Hawaiian populations, we've done lots of surveys up in Hawaii. And the only time we've ever found dimorphism in Hawaii, so most of the time these are all co-sexual hermaphroditic populations. The only time we find dimorphism in Hawaii is in 3 locations in Maui.
Okay.
Okay, but all of those populations are diploid. So we're pretty confident then that in Maui, when you get the evolution of dimorphism, it's happened on a background of diploidy, which is not that surprising, right? Lots of plants evolve dimorphism on never have a ploidy change.
Yeah.
So it's not, I mean, it's just different than, say, the model that I proposed, but the model I proposed was just a model, right?
Yeah, totally.
But what you also see is in Mexico, you do have tetraploidy, and we do have the ability to actually know now that it looks like they're self-compatible. And so that actually does look like the model that I proposed. And so what I'm saying is I think it's like the closer you look, the more variation I see. And so within this single taxon, you have polyploidy that leads to the evolution of dimorphism, and you have just dispersal that makes that mating system transition that then leads to dimorphism as this backup. And so it's just awesome. It's just really, it's really variable and powerful, right? Because it's not even like you need a sister taxon anymore. You have it all. You've got one lineage of Elysium Carolinianum with all this variation in it.
That's cool. Do you think Maui, it occurred 3 times, or do you think it's close enough that it was like one instant and then?
That's a good question that I don't know the answer to, but I suspect it's one event. These are so in Hawaii, where you find Lycium are all the points. So they're just growing on the lava right on all the points. So you don't typically find it inland in Hawaii. And as the crow flies, those are not very far from each other, right? It's on the northern part of Maui. But I do think some more surveys, wouldn't that be great? More surveys of Hawaii are always needed.
Yeah. Does anybody else have a question? Jenna says, great talk, Jill, very interesting plans. Thanks for a very clear explanation of complicated situations. Is there anybody else has a question? If not, I'm sure you could contact Jill later if you're watching this later and have something to ask. Oh, Pat Reeves has a question. So can you resynthesize the polyploidy?
Oh, that's a good question. I haven't tried. I think I probably could. So when I first started my job at Amherst, I thought, oh, what a great idea. I'll get my undergrads, we'll we'll use colchicine and we'll take cuttings. I think that's how I would do it, is these things root pretty well from cuttings. And so maybe that's the way to do it. If you have other ideas, I'd love to hear them. And then I was pre-tenure and I thought, well, maybe I shouldn't have my undergrads using colchicine. Honestly, it went through my head. But I do think that would be a pretty fascinating project to take on. You'd have to choose your species pretty carefully. And I think I might choose Elysium Carolinianum because it is the one that actually grows really well in the greenhouse. And so I could take a bunch of cuttings, root them with some colchicine, I think, and then see what happens. So take those diploids and then see what happens. It's a great idea. Are there other ways to do that? I would love to know if that's the case.
I'm not sure, maybe somebody will know. But do you find that they're all like Aluploides or autoploides? Do you know? Or is this like a can of worms?
I don't know. It's a can of worms. I don't know, but I suspect it's a mix. We've got tons of sequence data, so you would think I could sort this out a little bit better than I perhaps have. I think Lycium californicum looks like an autoploid. When you look at the genes, when you look at the way the alleles sort across taxa, and I think that there are some cases in Africa where you definitely have autopolyploids. So there's, I think, some good evidence for autopolyploides, sort of strict autopolyploids. That said, I do think there's a lot of allopolyploidy in Lycium, and I think a lot of hybridization. And so even with this new RADseq data set, I'm very cautious and trying to be careful about how to understand the evolution of these hybrids, because I do think that's a big issue in South Africa. I mean, our rad trees are giving us pretty good resolution, but part of the problem is I think we have just so much heterozygosity in some of these plants because they are polyploids and they're fixed heterozygote. You know, I think that's caused some issues, I think, in our radseed trees. So yeah, a bit of a can of worms, but still trying to work on that a little bit.
Yeah, awesome. I mean, so much to study. It's always kind of exciting. Well, thank you so much for coming and talking to us today, Jill. People who are still here, you can tune in next week for Mario Martin, who's going to be studying the evolution and functional significance of anther cones and buzz pollinated Solana. All right, thanks everyone.
Bye-bye, thank you.